say, I will figure out when during the finals week I will figure out when I can be available so that you can pick up your Scantron. If not, if you don't pick them up, I'm just going to, I prefer to just give them back to you on the day that you take the final. But if you're in a hurry and want to look at your Scantron, then I'll figure out when I can make myself available. Remember the final is worth 1.5, so it's 150 points instead of 100 points. So that hopefully you do well, and then it'll see the grades up a little bit. Okay? So let's go on to the last set of lecture here. The first 13 to 14 slides I'm going to breeze through for a couple of reasons. One is that I think 100% of it is a review, okay? And then you were required to look at them ahead of time, right? So hopefully this isn't the very first time that you see this. This set of exam is about cell division, basically, and then at the end we'll look at wound healing as a process, as an example of a process that ties everything, every single thing that we talked about in this class together. So for when we talk about controlling of cell number, okay, these are the topics. So we'll talk about mitotic cell cycle. We'll talk about how the number of cells in your body are regulated, and then the quality, and then we'll talk about how the mitotic cell division is regulated. Okay? So the regulation of the regulation. So in terms of numbers, okay? So I'll slow down just a little bit. In terms of the number, we have, each of us, human beings, have between 14 and 140 trillion cells. Obviously a smaller person would have closer to 14 trillion cells, and a much bigger person would have closer to 140 trillion cells, right? But you look the same way day after day after day, right? And if you're going to grow or shrink, it's a slow process, right? So you don't yo-yo like this. So therefore, the rough number of cells in your body are kept constant, is the idea. However, you every day, as you're sitting right there right now, you have cell proliferation or cell division going on. So, but you're not growing. It's because that is balanced by cell death, programmed cell death, right? So to keep the number of a cell population constant, then is a combination of cell division and cell death. And we talked about cell death. There are two types: necrosis, which is premature cell death, versus programmed cell death. Mainly, we'll focus on apoptosis. Okay. And we're not just talking about an individual. We talk about everything that goes all the way down to the tissue and organ level. Your tissues and organs don't yo-yo up and down like this. And also, we're also talking about a population, a community also, right? So 30 years ago, the population of the world was something like, 3 billion people or so. Now it's 7 billion. So what does that tell you about the number of birth versus death? That the rate of birth is higher than the rate of death is basically what you're talking about, right? That's why it's growing. So anything that grows, then there's this imbalance of one rate versus the other. Okay? So that's what we're talking about. In terms of your body, a couple of examples. Why would you have cell death? Right? Why would cells in your body are told to die? For instance, during embryogenesis, during the development of an embryo, or even a postnatally, a certain target cell may be controlled by multiple neurons, nerve cells, right? But at some point, you want more specific controls. So you want a particular target cell to be controlled by only one neuron. So therefore, out of the 2 or 3 neurons that are controlling that target cell, everybody has to go. Everybody has to die except for that one cell, right? So then what does that tell you about development? It tells you that cell death is important in refinement of activities, right? So as you grow, you don't just most of your neurons, you will not develop more neurons. In fact, your neurons will be dying. Okay, so that's one example again for refinement. For instance, for motor skills. If you want, as babies, you don't have fine motor skills, and one of the ways to have fine motor skills is to get rid of some of the neurons that are innervating, multiple neurons that are innervating the same cell. Okay. The other example is of apoptosis is that all animals, that vertebrates anyway, during embryogenesis, as a fetus, we all have webs in between the digits, similar to a duck. The difference is that for a duck, the web doesn't go away. For us, it goes away, right? And the webs in between the digits are made up of thin layers of cells. So then, as we develop into a human, postnatally, that all the digits are free, is because the cells within the webs are told to die through apoptosis. There are people who have a condition where they have webs in between their fingers. It's because something is wrong with the apoptotic process. Okay, so apoptosis is a necessary part of life, not just to get rid of old and dying cells or unnecessary, well, obviously unnecessary, but old and dying cells. Just a pretty picture about cell division, okay? So when we talk about the cell cycle, we're talking about mitotic cell division, okay? We're not going to, I'll mention mitotic cell division a little bit, but we'll talk about mitotic cell division. Mitotic cell division is only one of many different types of cell division, okay? Anyway, we have learned, this is an older picture, we have learned to control, to vary the rate of cell division and cell death. And if one outweighs the other, this is what you get. Okay, this is a bull that was on steroid, literally on steroid. And there are there are also natural occurrences where the rate of one, in this case is cell proliferation, outweighs the rate of cell death. Okay, and you get cancer. Okay, so again, if the two rates are imbalanced, then you get a problem. You get something different. Okay, so for the most part, you want the two rates to be in equilibrium. All right. So a little bit about cell division. Mitotic cell division is only one type. In fact, most of you have learned about the two types of cell division. One is binary fission. Binary fission is a type of cell division used by prokaryotic cells for bacteria for the most part, right? So divide into two. So that's binary fission. This is mitotic cell division here that most vertebrates use. There are other organisms that use mechanisms that are in between binary fission and mitotic cell division in terms of complexity. So binary fission is really simple. That's the simplest means to divide. Mitochondria divide through my binary fission, so quite simple. And mitotic cell division is extremely complex, which is why we learned it in this class. And a lot of other organisms have characteristics of both binary fission and mitotic cell division. What do you need to know about this slide? Just what I said. So don't try to memorize anything else except knowing that there are many, more than two, probably half a dozen different mechanisms for cell division. Okay? Okay, so what is the function of mitotic cell division? It is used to control cell number, but not just cell number, but control cell quality. Is that cell that is being produced a good cell? If it's not, your body won't produce it. And then cell type, and then we'll talk about differentiation. So we'll talk about stem cells. Okay. So mitotic cell division. These are the effector functions or the functions of mitotic cell division. Again, you've seen this slide about, I think this is the third or fourth time you see it. In terms of controlling cell number, first of all, for cell division, there are many signals, right? In order to tell the cell to divide, there are many signals. In order to tell the cell to die, there's only just one. Okay? And cell death has to do with controlling cell quality. Differentiation has to do with controlling cell type. And then controlling cell number is really the balance between cell proliferation and cell death. To bring in some of the things that we talked about before, signaling is important. So I just said that in order for cell proliferation to occur, then there has to be multiple signals. And remember the slide that I'll show again here, but in order for the cell to divide, there have to be at the very least 4 signals, right? One would be from a growth factor hormone to tell the cell to divide. And another one would be a signal to indicate anchorage. And another one is to indicate whether or not the density is too dense to divide. And the last is a signal to say that there's enough nutrients. So the very first one, the growth factor that tell the cell to divide, is called a mitogen. So a mitogen, by definition, is a signaling molecule, is a ligand that induces a target cell to undergo cell proliferation. And so normally, not always, but normally the mitogen then would activate a receptor that ultimately activate the MAP kinase cascade. So the MAP kinase cascade is used extensively during cell proliferation. Okay, and the MAP kinase cascade then would activate a certain transcription factor or transcription factors to regulate gene expression. So in order to tell the cell to divide, certain proteins have to be produced. Normally, if the cell is not dividing, those proteins aren't present. So that's how the cell know whether to divide or not. Okay? A reminder, and we'll go into more details about the signal transduction pathway. Right now, a lot of this is a reminder of what you should know. Here's the cell cycle. The cell cycle is composed of the GAP1, S, and G2 stages or phases. And those 3 constitute the interphase. And then the last bit is the mitotic cell division phase. In the M phase, it's actually, if you would do this, I'm going to ask you to add and correct several of the things that are on the slide, and I'll explain why that is in a minute, but let me do this first. Write mitotic cell division. It's just not mitotic phase, it's mitotic cell division stage, okay? Because mitosis has a different meaning than mitotic cell division, right? Mitosis is a part of the mitotic cell division, okay? So this whole phase is the mitotic cell division phase, or the M phase. Okay. So I'm going to be making a number of changes, additions and changes for the following reasons. Number one is that we've got, I built this with another instructor, the PowerPoint, and we didn't obviously, well, maybe not obvious, but we didn't draw these pictures ourselves. We got them from various sources. And sometimes, believe it or not, the sources that you get, the information that you get, even from textbooks, are incorrect or incomplete. Okay, so I'm here to tell you that what is incorrect and what is incomplete. That's number one. Number two, I'm going to be correcting certain things and add certain things to make things clearer for you. Okay, a lot of the processes are kind of muddy, so I define them and separate processes even more for you, so that hopefully makes it easier for you to understand. Thirdly, like I said, I build this and teach it with another instructor, and we teach things differently. And so the way that I work is that when I work with somebody, I usually let that person do whatever that person wants to do, and then when it comes time for me to teach the class, I just make the changes myself instead of fight over the details that have to go on the PowerPoint. There's no point, right? So those are the various reasons. So I'll be again asking you to make additions as well as changes throughout this PowerPoint. Just as a reminder, we'll go back and do this again. You need to know this, but basically what gap 1, what each of these phases are is, gap 1 is the period where the cell is building up, making more material in preparation for DNA replication, for the S phase. That's basically it. So if you notice, all of these things are required for DNA replication. And then S phase is for DNA replication. And gap 2 is the period where the cell, again, makes material, prepare things, getting ready for the mitotic cell division. That's basically it. Okay, so these are just examples, but examples of those big ideas. A reminder that that during mitotic cell division, you have prophase, prometaphase. I usually lump prophase, prometaphase into prophase. So prophase, metaphase, anaphase, and telophase. That part is mitosis, right? Mitosis includes prophase, metaphase, anaphase, telophase. And then mitotic cell division includes mitosis and cytokinesis. So then cytokinesis is included. What happened is that the cell would go through prophase, metaphase, and then anaphase is initiated, but as soon as anaphase is initiated, cytokinesis is initiated. So anaphase and telophase overlap with cytokinesis. And I'll explain all of the details, as much detail as necessary in a few minutes here. Okay? So again, prophase, metaphase, anaphase, telophase. And as soon as anaphase is initiated, cytokinesis occurs. Okay? And these are the various things that have to occur. And again, I'll have pictures and I'll describe all of these processes as we go through the slides. Pretty pictures. This is metaphase, remember. This is anaphase, right? And so to get to metaphase, if you remember, sister chromatids have to be pushed toward the spindle equator or metaphase plate right here, right? And once they're at the metaphase plate or spindle equator, then that's metaphase. And the sister chromatids are pushed toward the metaphase plate by the assembly of microtubules. Okay, and then they get pulled. The sister chromatids are separated and pulled toward opposite ends by, and you probably learn, and I probably teach at the Bio 2 level, they are being pulled through the depolymerization of microtubules. That's not quite correct, right? Even though we teach that way at the lower level, at this level, you need to understand more detail. So I'll tell you why that's not quite correct. But in general, that's kind of and sort of true that while the sister chromatids are being pulled apart, depolymerization of microtubules occur. Okay, but I'll tell you why that's not completely true. So moving from metaphase into anaphase, that's a big deal. Okay, sometimes a cell stays in metaphase for a long time and won't move on. So if a cell is told to move on, that's a big deal. Okay, so it takes a lot of regulation to tell the cell to go from metaphase to anaphase, and we'll talk about that. Okay, so we'll talk about the regulation of going through the cell cycle. So here's what happens. Here's where it's not really the depolymerization of microtubules that pulls the sister chromatids apart. Here's what happens. Here's a sister chromatids, the X right here. There's a kinetochore that's found on both sides of the sister chromatids, right? And the kinetochore is connected to, associated by a molecular motor, dynein. Okay? And the dynein then interacts with the microtubule. So what does dynein do? Dynein walks along the microtubule toward the minus end. So here's what happens when, during the pooling and separation of the sister chromatids, is that as the dynein molecular motors walk along the microtubule toward the minus end, then the dynein motors pull on the kinetochore, which in turn pulls on the sister chromatids. So it's really, mechanically, it's really the motion of the dynein motors that separate the sister chromatids, right? However, if you have the microtubules here and the kinetochore and dynein here walking back, what happens at some point? The microtubules would inhibit that movement. So in order for the dynein to continue to walk, then depolymerization has to occur. So again, depolymerization has to occur, but it's not the process of depolymerization that pulls the sister chromatids apart. It's the activity of the dynein motors. Right? And it used to be a mystery. I remember when I first started teaching here 11 years ago, it was still a mystery. It wasn't until probably 2 or 3 years later that people figured this out. So we've only known about this mechanism probably no more than 8 years ago. Okay? So that's what happens. That's exactly what happens, right? Cytokinesis. Cytokinesis occurs because there's a contractile ring that is formed, and contractile ring is composed of microfilament or active filament bundled in the contractile bundle or loosely bundled, and then intercalated in the space in between with the myosin filaments. Okay? So when you have the contractile ring having both myosin and microfilament, and then the myosin and microfilament pull at each other, so contraction. So contraction is what causes the contractile ring to get smaller. And then as the contractile ring gets smaller, the microfilaments pull at the plasma membrane, and that's what allows the plasma membrane to be pulled in. However, again, the microfilaments are fairly stable at this point, right? Once the contractile ring gets to a certain point, it can't get smaller. So what has to happen? The microfilament has to get smaller, and that is done by severing. Not depolymerization, that's too slow. Remember the difference between severing and depolymerization? Severing occurs because you want quick disassembly of the microfilament. Okay? So 3 processes occur almost simultaneously. The contraction or the pooling between myosin and active filaments, the pooling of the plasma membrane, and severing of the microfilament occur as the contractile ring gets smaller, and that's cytokinesis. And then how can the plasma membrane, at some point, how can they fuse in to make 2 cells? What is that property that we learned about for cell fusion, membrane fusion? Is that the membranes have to have similar characteristics, right? Lipid signature. You can't have more similarity than the exact same lipid signature, right? So that's how. So by having the contractile ring get smaller and smaller and smaller, bringing the plasma membrane closer and closer, then at some point the plasma membrane is close enough to fuse in to itself, right? And that's what happens. So there's no need for an enzyme that would induce fusion. And that's a pretty picture. Cool. Plants do undergo cytokinesis slightly differently because there isn't a contractile ring. Right? It's not necessary to have a contractile ring. What the plant does is that it starts forming in the middle, forming a cell plate. The cell plate, all it is the cell wall. So basically, a plant will start forming the cell wall right in the middle during anaphase. And then as soon as the two cell wall from each of the daughter cell is completely formed, now you completed cytokinesis. That's it. Okay? So again, no need for a contractile ring. And there's no such thing as a contractile ring for plant cell division. Okay, review. Now we'll come on to new stuff about cell cycle, about the regulation of cell cycle. We should have plenty of time for 3 lectures is enough for the rest of the lectures. So I'm going to take 2 seconds out to see where we are. Are you comfortable with what I just said? Do I need to repeat anything? Yes, ma'am, and then sir. Yes, ma'am.
I think we said the 3 things that need
Cytokinesis is 3 events that have to occur simultaneously. Is the pooling between myosin filaments and microfilaments, or basically contraction of the contractile ring. And then the plasma membrane is being pulled inward is the second one. And then severing of the microfilament is the third. All 3 of those, if any of those 3 is missing, then cytokinesis can't occur. And yes, sir.
I was slightly confused when you described the severing process. So when they sever, is that just split the cells apart, or?
No, severing of the microfilament.
Right, so that's actually
Right, making the microfilament instead of this big, make it shorter, so as contraction occurs, that the microfilament doesn't push against the plasma membrane. That's good. Any other question? All right. Now is the regulation. Now it comes the interesting, I think, part. And I also am going to make additions and additions, okay, to the slide to really make it more easier to comprehend, okay? So, okay, I should have done this earlier, but that's okay. So what I want you to do is this, is right here, at this point right here, instead of having 4 checkpoints, I'm going to have 6 checkpoints, right? 2 of them are missing and 1 of them is altered, and I'll tell you why. And I promise you that how I do this is going to be clearer than what you see here. So before, the very first thing should be a G0/G1 checkpoint. And I'll explain what a checkpoint is and all of that in a minute. So that should be a G0/G1 checkpoint, and that really is. That goes with this definition, to enter the cell cycle or not. Okay? And then you have the G1S checkpoint. I will explain what that is in a minute. And then you have the S checkpoint, that's fine. In between the second and third one, you would have the SG2 checkpoint. That's something that most people don't pay attention to. There is a checkpoint there, people just don't talk about it. And then you have the G2N checkpoint and M checkpoint, and that's perfect, okay? And you see, moving from metaphase to anaphase is so important that they have that process has its own checkpoint. And a checkpoint is a huge thing, right? So there are 6 checkpoints again, G0, G1, G1S, S, SG2, G2M, and M, okay? And the change is that the G0, G1 actually checkpoint goes with this definition, and then the G1S checkpoint I have to explain. The definition is not easy, so I have to explain what that is. Okay, so what happens? What happens as a cell goes through the cell cycle? The cell has to be, number 1, told to enter the cell cycle, and then as it goes through the cell cycle, it wants to make sure that everything is okay, and we'll identify what okay means, right? If a cell is not okay, it won't be able to go through the cell cycle. So it has to be okay, right? And we'll define what that is. And then all the way to the end. So I have to go through all of these. And a checkpoint is just like a border checkpoint, right? So a checkpoint to go from one region to another. Just happens that these checkpoints aren't physical checkpoints. They're abstract checkpoints. They're not even chronological, they're not even a concrete time checkpoint. It's not like it's 3 hours and then it jumps to the next checkpoint. It's more abstract than that, and I'll explain what checkpoints are. But before I talk about all of that, what is G0 and what are all these phases and what does all this mean? Let me move back. Let's see. I'm sorry. Oh, okay, here. Okay. And again, as you can see here, some books has 3, 4, 5. I have 6 checkpoints. So checkpoints depends on who's talking, what you look at. People have different checkpoints. So I'll explain everything. So during the interface, you have G1, S, and G2, right? And that means that a cell is in the cell cycle, going through the cell cycle. What happens if the cell is not going through the cell cycle? What checkpoint? What phase of the cell cycle is it in?
G0.
It's G0. It's not in the cell cycle. So G0 is not a part of the cell cycle, right? G0 stems like I draw a loop from G1 like this, right? That's G0. G0 or gap 0 is also called the quiescent stage, like quiescent stage. It seems to mean that it's not doing anything. In fact, a cell that is performing a function, like a liver cell that has to detoxify toxin in your body, right? If it's performing a function, it must be in the quiescent stage. A cell that is going through the cell cycle will not perform its specific function. Okay? So again, a cell that is performing a specific function must be in the G0 or quiescent stage. Otherwise, it's too busy going through the cell cycle. Okay? So that's what that is. So now, in order for a cell to be performing its function, and then now, let's say your body says that, okay, the liver has to produce more epithelial cells, more liver epithelial cells or hepatocytes. Then, while it's performing its function, it's going to be told to exit G0 and enter the cell cycle. So it has to go through a checkpoint. And so that's the G0, G1 checkpoint, is the checkpoint where the cell is told to re-enter the cell cycle. Okay? So that's what this slide calls the G1 checkpoint, which cross out. That's the G0, G1 checkpoint. Okay? And then there's no such thing, as far as I'm concerned, as the G1 checkpoint. And then you go to the G1S checkpoint is the cell has to ask itself, is it ready to move out of G1 and enter S stage? So that's a checkpoint, right? But here's the deal. Here's the deal. At some point, as the cell goes through the cell cycle, it's going to be told that it's too late to go back. Right? At some point. Not early on, but at some point, there's a point of no return. So if a cell, let's say right here, and it can't for some reason continue with the cell cycle because there's a lack of nutrients, guess what? It's going to be told to die. So at some point, right, there's a point of no return. We call it on the slide, it's the go ahead point. Some people call it the start point. It's capital S-T-A-R-T, all capital, right? Either start or the go ahead point. Okay? And that go ahead point or start point is between the G0, G1 checkpoint and the G1S checkpoint. So it's somewhere right here. Okay? It's not a checkpoint per se, and I'll explain why it's not called a checkpoint, but it is a go-ahead signal. And if a cell is told that to has that go ahead signal, then it will continue, and then it will go through the G1S checkpoint. So the G1, once the cell gets to the G1S checkpoint, that tells the cell that it has passed the go ahead signal. So that's why the G1S checkpoint is a little difficult to explain, is that it really doesn't do anything except to tell the cell to enter the S stage, right? But the cell that enters the S stage doesn't undergo DNA replication right away. The cell undergoes DNA replication only after it passes the S checkpoint. So that S checkpoint is in the wrong place. That S checkpoint should be right here. This is right here. Cross that out and write G1S checkpoint. So this is the G0, G1 checkpoint. This is the go-ahead signal or the start. This is the G1S checkpoint, and the S checkpoint is actually here. So the cell would have entered the S stage, and then won't start DNA replication until probably half an hour to an hour after it's entered the S checkpoint, S stage. Okay. So again, the importance of G1S checkpoint is just to make the cell realize that it has the signal, the go-ahead signal to progress. And so that go-ahead signal or the start point is the point of no return. If it hasn't gotten to that start go-ahead point, it actually can go back and enter the quiescent stage again. And I explained to you at the molecular level how that happens in like 10 minutes here. Yes, sir.
Well, you probably picked it, but are these lag periods timed to allow necessary enzymes and signals to go back?
Right. So at the same time, you have a lot of gene expression, you have a lot of reactions occurring. Yes, yes. So roughly, the cell will spend about 4 to 5 hours doing G1, about 9 to 10 hours doing G2 in S, about 2 or 3 hours in G2, and only about 1 hour for mitotic cell division. So and we'll talk about human cells. So for human cells, it takes roughly 16 to 24 hours to go through the cell cycle, and to complete the cell cycle, more toward the 16 hours than the 24 hours. Okay.
We've been told that these G phases are also growth phases. Is
Right.
G not
Growth. So
maturation of the cell?
growth meaning literally growing bigger. Yes. So yeah, in terms of making more materials, it's also making more phospholipids and add to the plasma membrane. How does it add to the plasma membrane? By vesicular transport. Right. So everything that we talked about before. Yes. So the cell grows. In fact, after a cell division occurs, right, the daughter cell is never the full size. It's slightly smaller. So during G1, actually, the cell has to grow to its normal size. And then G2 then would allow the cell to grow to roughly twice its size, not quite, but almost twice its size. So that when you have cell division, that the daughter cells aren't too tiny. Right? So the daughter cell won't be half the size of the parental cell. So you are right in that. And that's a part of the making more material. Yes, sir?
How about for G sub-zero?
Sub-zero.
Or just zero? G0.
G0, then there's no growth.
No growth.
Right, there's no growth. Now I already told you there are 2 examples of organelles that actually are produced during G0, which is mitochondria and peroxisome, right? Otherwise, no other organelle. Well, and the lysosome kind of sort of, because the lysosome is one of those, it's within the vesicular trend, within the endomembrane, so the lysosome gets bigger and smaller, and sometimes you remove it, and so that's slightly different. But for the most part, only mitochondria and peroxisome are being produced. But during the 2 gap phases, not G0, but gap 1 and gap 2, lots and lots of different materials are produced. And by the way, gap 0 is not a part of cell cycle. Okay, so I told you what the checkpoint is, and the reason why that go-ahead signal is not called a checkpoint is that by definition, a cell cycle checkpoint is regulated by 2 proteins or by a dimer, right? And that go-ahead signal is not regulated by that dimer. It's regulated by something else. So that's why it's not called a checkpoint, but it is a major point within the cell cycle. Okay, and I'll tell you what that dimer is in a minute. Yes, ma'am.
Which one did you say is not a checkpoint?
The go-ahead signal is not called a checkpoint, but it really is, right? It's actually more important than any other checkpoint that there is. Okay, so I talked about the G0. That I talked about the go-ahead signal, right? Okay. So let me. Okay. So let's see why is it that you have between the G0 checkpoint and the go-ahead signal or the start. Why do you need that? Why do you even need that? Why can't you just go through? It's because cell proliferation or cell division is activated by multiple signals that the cell may or may not receive at the same time. But think about the signals that it's not immediate. So if a cell receives a signal from a growth factor, a mitogen, right? So the mitogen, this is where that signal is, to tell the cell to undergo cell proliferation. How long does it take for the cell to actually build up and undergo gene expression? Quite a while, right? A few minutes at least. Maybe even an hour or so, right? And at the same time, you might have received information about anchorage dependence. But again, that means that the integrin might be activated, and that sends a signal into the cell to say that, hey, there's anchorage, but how long is that going to take? That takes a little while. And if there isn't anchorage dependence, that also takes a while. So the cell, while it's going to go ahead and prepare for cell proliferation, it doesn't know whether or not there's anchorage dependence, right? It doesn't know whether or not there are too many cells around because that information hasn't come in yet. So the point of G0, G1 checkpoint is only due to the mitogen here, right? The cell is told to go ahead and enter the cell cycle, but again, it hasn't received other signals. So that explains why there has to be a go-ahead signal. So that between Z0 and that go-ahead signal at the start, that's when the cell has to accumulate enough information to see if it truly can continue with the cell cycle. Does that make sense? So that's why that go-ahead signal is important. And you've seen this slide before, so there's no reason for me to explain that, right? So checkpoints. Checkpoints are controlled by something called a cyclin-dependent kinase. It's a kinase, it's a phosphorylate thing. Okay, but CDK, all CDKs have to dimerize with a cyclin, and so the cyclin is a regulatory protein. Cyclin tells the CDK to function, right? And CDK is a whole family of CDKs. CDK 1, 2, 3, 4, 5. Cyclin, there's a whole family of cyclin A, B, C, and then different combination of cyclin and CDK then regulates different checkpoints. Okay? And again, we know that instead of G1, that actually should be a G0, G1, and then G1S, and then S, and then in the middle you should have the G2 checkpoint, and then you have the G2M checkpoint, and then the M checkpoint. Okay? So you notice, and don't worry about this just yet. I'll explain everything, so hopefully it'll be clear. You notice that we're talking about cyclin, regulation of a cell progressing through the cell cycle. We just talk about expression of cyclin. We don't talk about expression of CDK. Okay, and there's the reason why. Okay, so this slide is important, but it'll make sense later on. All right, but right now, what you need to know is that a checkpoint is regulated by the CDK cyclin dimer. That's really all that you need to know right now. Okay, and when CDK is activated, the particular CDK will be used to phosphorylate something. Okay, now here this is where my adding extra checkpoints. This is where it helps. So, for instance, the G0, G1 checkpoint is controlled by CDK4, CDK6, and cyclin D. and then you have different combination. Don't write that down. That's way too much, right? I spent 30 years trying to memorize all these numbers and letters, and it's not necessary, right? Because you're not doing research in this area, so you don't have to do that. So then, if I ask you which CDK and which cyclin controls the G0, G1 checkpoint, you would say it's the G0, G1 CDK and the G0, G1 cyclin. Simple enough, right? So if you know the exact checkpoint, then that's the name of the CDKs and cyclin. That's all that it is. Because it will get more complex, because G2M checkpoint and the M checkpoint, guess what? It's controlled by the same CDK and cyclin, but they do slightly different functions. So they're controlled by CDK1 cyclin B, right? But they do different functions. So then, if I say G0M, G2M checkpoint, G2M CDK cyclin dimer, then you know that it performs a couple of functions, I'll tell you. And then if I'm referring to the M CDK cyclin dimer, then you know that it's the other two functions that I will tell you, right? Even though, again, they're the same complex, okay? So again, by knowing the exact checkpoints, it's easier to name and understand these processes. All right, so here's how regulation occurs. You remember, actually, let me do this first and then go back 2 sides. So that's the mitogen again, activating an RTK, which activates the RAS, which activates the MAP kinase cascade, which activate a transcription factor, which activate gene expression. Right? Gene expression of what? Gene expression of proteins that are important for cell division. Gene expression of all, not just one, but all of the CDKs that are important for cell cycle. Every single last one of them. Whether it's the G0, G1 CDK, or it's the SG2 CDK, or the M CDK. Right? All of them will be produced at the beginning. Once the cell enters the cell cycle, all CDKs are produced at lower amounts, but not all cyclin. So at this point, only G0, G1 cyclin is expressed. And so in order for cell, so I'm hoping that you're getting the picture here, in order for the cell to know that it's which stage it's in is by the presence of specific cyclin. All right? So if the S cyclin is, so there's always a production and there's always a lag period and there's a peak, and then eventually the cyclin goes away. So if there's a peak of the S cyclin, then you know that DNA replication is about to start or had just started. Okay? So that's for the most part, the major means of regulating the cell going through the cell cycle is the production of cyclin. And something about cyclin is that, number one, it is a regulatory protein to regulate CDK activity. And number two, cyclin has a short half-life. So you don't want the S cyclin to be hanging around when you're in the M stage, right? You don't want it, right? So you can see why the cyclin is there. But CDK is going to be there throughout. Okay. Did you have raise your hand? Okay. Okay. Good so far? So, again, this is the G0, G1 CDK. Okay. Meaning that it has, even though I write CDK, but it's CDK cyclin dimer. And this is G2M CDK and M. Remember, so G2M CDK actually performs these two functions, and the M CDK actually performs this function and one more. So one is missing, and then again the G2M CDK performs this function, and the M CDK performs that. Again, still the same CDK and cyclin, but it's regulated differently. Okay, and thank goodness we're not going to go into the regulation of the regulatory proteins. That's even more complex. Okay, so here's what happens: you get a signal, and the cell is going to be told to produce all the CDK. Great. And the cell is also going to be told to produce the G0, G1 cyclin. Great. The G0, G1 cyclin binds to the G0, G1 CDK to form a dimer. What does that dimer do? That dimer phosphorylates the RB protein. So here's what happens in the cell during the quiescent stage or the G0 phase. RB protein in the unphosphorylated form has very high affinity for an important protein called E2F. Right? Only in the unphosphorylated form. If RB protein is phosphorylated, it no longer has affinity for E2F, and now E2F is released. And E2F, when it is free, it actually is a very strong transcription factor. So all the CDK as well as not all the CDK, but so far okay, never mind. Okay, so far so good. So E2F is a very strong transcription factor. What does it do? It induces the expression of the G1S cyclin as well as the S cyclin. So this is missing right here. So G1S cyclin and the S cyclin. So here's, at the molecular level, here I'm about to explain the go-ahead signal, right? The start. How does that happen? Okay. So again, G0 normally in the quiescent stage, unphosphorylated RB protein binds to E2F. So no cyclin is being expressed. Well, no G1S or S cyclins are being expressed, right? Now mitogen activates MAP kinase cascade, activates gene expression of all CDK, as well as the expression of the G0, G1 cyclin. So far, so good. And then the two combine to form a dimer. And that dimer then controls the G1, G0 checkpoint. How does it control it? By phosphorylating RB protein. That's the control, right? When we say checkpoint, that's what it means, is some kind of a reaction is occurring. Okay? So in order for the cell to go from G0 into G1, RB protein has to be phosphorylated. Now between RB, right, between that phosphorylation and the go-ahead signal, as well as onto G1S checkpoint, here's what happens, is phosphorylated RB protein would release E2F. E2F then will induce the expression of G1S cyclin first, and then a little bit later S. When G1S cyclin is expressed, what happens? It will dimerize with the G1S CDK, which then tells the cell to move from G1 into S. Right? And then when S is expressed, S then would dimerize with the S CDK, S cyclin would dimerize with S CDK, which will tell the cell to initiate DNA replication. So here's where the go-ahead signal occurs. What happens if RB protein is phosphorylated, and then at this point the cell receives information that, hey, there's no anchorage, right? Then the cell will carry out dephosphorylation of RB protein. When RB protein is unphosphorylated, what does it do? It binds to E2F. If E2F is bound, and E2F is no longer able to carry out gene expression, then the cell this is when we say that the cell goes re-enter the quiescent stage. Okay? Does that make sense? So that's the go-ahead. The go-ahead signal is if E2F is not only released by RB protein, phosphorylated RB protein, but would be able to induce the expression of G1S cyclin at least. Once that happens, then that's a point of no return. Once G1S cyclin is produced, that's a point of no return. So far, so good? Okay, and you see why multiple signal is important, and you see why the timing is important, I hope. So then, I know some jokers is thinking it right now, and they're saying, well, what happens if you have let's say 10 E2F free, and 1 or 2 E2F is already made its way into the nucleus and then induce expression of G1S, but then not all of them will go in there, but then RB protein is going to be dephosphorylated, and then the rest of the E2F is going to be bound, right? Then does the cell go on? Does the cell move back into quiescent stage? What do you think? Threshold. It's always about threshold. It's never about 1 or 2 molecules. It's never about anything concrete in cell biology. It's about threshold. So there has to be a threshold amount of free E2F in the nucleus, and there has to be a threshold amount of G1S cyclin that is being produced, right? So once that threshold is reached, then that's the point of no return. Any point before that, you can have a few molecules of G1S cyclin produced, and still the cell hasn't gone over that go-ahead signal. Does that make sense? So it's not about any concrete number. It's about the threshold, right? It's not about 1 or 2 molecules of G1S cyclin being produced. It's about there has to be enough G1S cyclin produced that increases the chance of what? Of G1S cyclin binding to G1S CDK, right? To do what? To have enough of those dimer, right, to get the cell to go from G1 into S stage. So there has to be a certain threshold. Yes? No? Okay, that's tiring. I hope you understand that. I see a few nodding heads, so that's a good sign. Thank you. Okay, so that's that. We're going to skip past DNA replication. Not going to talk about it at all. We're going to skip past the SG2 checkpoint, even though I made you add it in, but now we're not going to talk about it. Now we're in gap 2, and now we're preparing to enter M stage, right? And so then the G2M cyclin is going to be produced, dimerizing with the G2M CDK. So what I'm going to do is this is going to talk about the molecular aspects here, and then try to overlap it with the physiological aspect of the pulling and pushing of the cystochromatins. Okay. So the first thing is I'll tell you the function, the mechanics, the molecular aspect, the mechanics of the G2M CDK cyclin dimer. What does it do? It phosphorylates histone. To do what? To change the histone code. What happens when the histone code is changed? It changes the histone code in a way that tells the cell to undergo chromosome condensation. What is one of the characteristics of prophase? Chromosome condensation, right? And you learn that, but how are they told to condense? By phosphorylation of histones to have a histone code to have a certain histone code to tell the cell to condense. Not just into the heterochromatin structure, but even further. Okay? So that's one thing that the G2M CDK cyclin dimer does. The next thing that it does is phosphorylating lamin. We learned what lamin is. Lamin is a subunit of the nuclear scaffold, which is a type of intermediate filaments. Lamin in the unphosphorylated form have affinity for one another to build its nuclear scaffold. What does the nuclear scaffold do? It holds up the structure of the nuclear membrane, right? So when lamin is phosphorylated, what happened to the nuclear scaffold? It breaks down because lamin no longer has a phosphorylated lamins don't have affinity for each other, for one another. All this is complete review from what you learned before, right? What happens when the nuclear scaffold breaks down? it no longer is able to hold up the nuclear membrane. So what's another characteristic of prophase? The disintegration of the nuclear membrane, and that's how it happens. Is the breakdown of the nuclear scaffold causes the nuclear membrane to disintegrate. And the nuclear scaffold is broken down, breaks down due to the phosphorylation of lamin. Lamin is phosphorylated by the G2M CDK cyclin dimer. Very good. And now we're in prophase, and microtubules will so then going toward metaphase, right? Microtubules are going to undergo assembly, and from the two poles, and then pushing the cystochromatids. And what the microtubule does is that it grabs a hold of the kinetochore, and then as this assembly occurs, it pushes the cystochromatids toward the other side, right? Well, the cystochromatid at this time, each cystochromatid has two kinetochore. But at this time, only one kinetochore is interacting with the microtubule. What about the other one? It's free. Okay? So what happened is that there will be another spindle fiber on the other side that is undergoing the assembly, and here's the important stressing this point, is undergoing the assembly at the same rate. So as this spindle fiber pushes the cystochromatids and undergoing assembly to push the cystochromatids, the other spindle fiber also undergo assembly at the same rate so that they would meet in the middle. So that the other spindle fiber would grab a hold of the free kinetochore. This is why there is absolutely no control, no reaction, no enzyme that control how cystochromatids line up along right smack in the middle of the cell in the metaphase plate, right? There's no enzyme that controls that. The only control is by the fact that spindle fibers assemble at the same rate. Same with anaphase, where the cystochromatids are being pulled apart and they end up at the opposite pole at around the same rate, is because disassembly occurs at the same rate. That's very important, right? Yes, sir?
So then how is that regulated through like the enzymes that assist in assembly?
That makes sense.
Like having the same concentration
Right, so
each end, how is that controlled?
very good. So therefore, the cell has to ensure that there is an evenly distributed concentration of tubulin and make sure that stamen is phosphorylated and everything. Yes, so the cell still has to control the rate that way, but it's more of even distribution instead of one or the other. Okay, so that's an excellent question. All right. Let me go on. Anaphase. Anaphase, and there are two other slides for this. So I'm just going to say that the MCDK cyclin dimer regulates the cell to progress from metaphase to anaphase. Right. So that's the part that is not here. So it's an MCDK cyclin does that. As soon as the MCDK cyclin dimer tell the cell to progress from metaphase to anaphase, which again I'll tell you the detail in later slides, the same MCDK cyclin also phosphorylates myosin to tell the cell to undergo contraction within the contractile ring to start cytokinesis. So the building of the myocontractile ring is just to prepare for cytokinesis, but cytokinesis won't occur until the contraction occurs, right? So the formation of the contractile ring is not a sign of cytokinesis. It's the contraction and the contractile ring getting smaller. That's the sign of cytokinesis. So again, that same AMCDK cyclin that activates the cell to progress from metaphase into anaphase also phosphorylate myosin to initiate contraction of the contractile ring. Yes, sir?
So both of those would be required?
Required, right,
So
yes.
is that a form of crosstalk at all?
and they're not required. There are divisions where cytokinesis doesn't occur, and I'll show you examples of that later. Okay. We're good? All right, we'll start the slide after this.